Thank you. Thank you for the invitation. So I will tell you today how we're dissecting the cellular landscape of the Alzheimer's brain. And more generally in my lab, we're studying the cellular and molecular changes in aging brains and that are underlying age-associated neurodegenerative diseases. So as you all know, we're living in a society where the aging population keeps increasing even during these times of pandemics. And the aging-associated neurodegenerative diseases has become a huge burden in our society since we don't have any effective treatments or prevention strategies. So that's a really great motivation for us to try and understand better these diseases and try to understand them from a different perspective than what has been so far been done. Specifically, we've been focusing a lot on Alzheimer's disease, which is a progressive neurodegenerative disease. I think you all know Alzheimer's, since it's so prevalent in our society, it's actually the most common form of dementia. And at the clinical stage, there's severe memory loss and cognitive decline. And at the later stages, the damage to the brain is so severe that we have, it leads to death. When we try to understand at the molecular level, we actually know a lot about this disease, but we also don't know enough. So we know that there is accumulation of abnormal protein that aggregates and accumulate in the brain over a long period of time. And these protein aggregates, so specifically amyloid beta plaques that are extracellular and neurofilament tangles that are intracellular, they are leading to damage to neuronal cells and death of neuronal cells that eventually we believe are the cause of the cognitive decline. Yet when we're looking at the last 450 clinical trials for Alzheimer's disease, we're seeing that a lot of them have been surrounding the amyloid beta plaques. We see that almost none of them have proven to be effective, which really calls for a different view of how we're studying Alzheimer's disease. And moreover, we know that Alzheimer's disease is now starting multiple years before any signs of cognitive decline. So it suggests that we should look at changes that are occurring before the severe loss of neuronal cells, other changes that occur in the brain that might be early drivers of the disease. So this is what we're doing in the lab. We're doing in the lab, and we're transitioning from a more neuronal-centric view surrounding the disease pathologies to a more holistic view looking at the entire cellular environment of the brain. So if we look under the microscope in the brain, we wouldn't see this nice, clear neuronal network, but in fact, we'll see a highly complex cellular environment where neuronal cells are surrounded by multiple other cell types that are all really critical for the function of the brain. So we have microglia cells, the resident immune cells of the brain, oligodendrocytes that are forming the myelin nerve cells, astrocytes that are important for homeostatic and metabolic balance in the brain, vascular cells, and so on. And all of this complex cellular environment is really, and the communication between the cells are really critical for the function of the brain. And we now know that there are evidence that all of these cell types are actually changing during Alzheimer's disease. And our hypothesis was that they're not only changing as a response, but rather this changes occurs early during disease progression is an important driver of the cellular cascade that is leading to cognitive decline. So we want to understand what is this unique cellular environment underlying Alzheimer's disease. So this has been our goal over the past few years, and the way we've been trying to address that is by building detailed cellular maps of the brain in healthy brains and Alzheimer's brain, and in order to understand what are the cellular and molecular drivers of disease. And we've been using the single nucleus RNA sequencing technology I've developed at my time in the Broad Institute to profile every individual cell and track individual cells during this progression of the disease. So we're not the only people using single nucleus and single RNA sequencing to profile the Alzheimer's brain. And one of the stars that have emerged from these previous analyses has been microglia cells, which are been switching to a disease stage early during disease progression. We're still searching for their function in disease. What I'll show you today is how in my lab, we're actually starting from profiling tissues using all kinds of omics approaches like RNA sequencing, then building these detailed cellular maps and going back to the bench to do functional validations. But today my talk will mainly focus on our more computational aspects of our work. So I'll start by showing you what we learned so far by building cellular maps of mouse models about disease-associated glia cells in the brain, then transition to talking more about the human brain and how we're starting to identify the cellular environment of the brain by multicellular communities. So in mouse models, our work has been in collaboration of multiple labs, especially Michal Schwartz's lab from the Weizmann Institute. And we've been profiling wild-type mice in an Alzheimer mouse model called 5XFAD and profiling the hippocampus and the cortex brain regions of these by single nucleus RNA sequencing, profiling hundreds of thousands of cells. And these are the maps that we've been generating of the hippocampus, where you can see on the left the healthy mice and on the right the Alzheimer mouse model. And the different cellular populations we've captured have really profiled the diversity of cells we were expecting to see in the hippocampus brain regions, multiple subtypes of neuronal cells, and all the different types of glia and vascular cells that we were expecting to find. So overall, on a bird's eye, you couldn't see a dramatic change between healthy and Alzheimer's disease, but we could already identify the disease-associated microglia that appear in Alzheimer's that was previously found. But one thing that really kind of struck us from these more detailed, unbiased maps was a very dramatic change we could see even by eye in astrocyte cells. So we're very interested in profiling them, and we're focusing our analysis in later stages mainly on these cells. So what are astrocyte cells? I'll just remind you that these are cells that have multifunctional cells in the brain. I think they have the most functions out of all the other cell types. They're important for homeostasis of the brain. They're participating in neuronal function. They're maintaining the integrity of the blood-brain barrier. They're important for metabolism of neurons and other cells, and they already are known for their important role in brain repair. But for some reason, they haven't been studied that much in the context of Alzheimer's disease, even though Alzheimer's himself, Dr. Alzheimer's himself, has identified them as turning into a reactive state called gliosis at an early stage of the disease. So when we focus on our analysis on astrocyte cells alone, we could now use diffusion map analysis to look at their transition across different cell states, since these are not discrete populations, but rather a diversity of states along a continuum. So you can see on the left the wild-type brain, we can find a range of states from a homeostatic state to a GFAP high state, which is a marker of reactive astrocytes. And what happens in the Alzheimer's brain is we see a completely new state appears, which we call disease-associated astrocytes. And these new cells that are really highly specific to the Alzheimer's brain are seem to be not only that they appear, but we also see a decrease in the homeostatic astrocytes. So when we look at the richness of our data, we could identify that disease-associated astrocytes upregulate a number of really interesting pathways, inflammatory-related pathways, many metabolic-related pathways related to lipid and cholesterol metabolism, and also other stress response like reactive oxygen species response. We also took markers of our cells and stained the brain to showing that the disease-associated astrocytes, they're not randomly positioned, but rather strongly biased towards in proximity to Alzheimer's disease pathology. So overall, we have really strong evidence that they have an active role during disease, but we wanted to try and put this on a timeline. So we were profiling the Alzheimer's brain when there's already cognitive decline and neuronal loss. So we then repeated our analysis looking at earlier time points and later time points. And when we do that, we see a constant increase in disease-associated astrocytes throughout disease progression, and a constant increase in the homeostatic state of astrocytes. And this suggested to us that there is a transition from a homeostatic state to a disease state. This was a little surprising, since actually disease-associated astrocytes are also reactive-like. They're much more similar to the common, to the physiological reactive state we saw also in wild-type mice. This and so we used some mathematical modeling to show that indeed in our model, we predict a transition from a homeostatic state to a disease state. So just to summarize before summarizing all our findings so far, one last important bit of information is that when we ask when do we first see the disease-associated astrocytes appear, we see them appearing early at around 4 months old mice, before any signs of cognitive decline and neuronal loss, but there is already the beginning of the Alzheimer pathologies at this mouse model. So this suggests that disease-associated astrocytes take an important role during early stages of the disease. Yet we have actually more questions than answers at this stage. So I'll just summarize what I've shown you so far. We found a very distinctive state in astrocyte cells that are not similar to the physiological reactive astrocytes that we see in normal brains. They're specific to disease. They have both linked to immune and metabolic signaling. They're positioned next to A beta plaques. They appear early before any signs of cognitive decline, and they increase as the disease progression. There's also some kind of mild signs that they might appear also with natural aging, suggesting an interplay between the genetic basis of the disease and the pathology to natural aging. But as I said, there are more questions than answers at this stage. We're still interested in understanding the functional role of these cells during disease progression, how they interact with other cells, and of course, the relevance to human brains. So I'll just show you more information from the mouse models before transitioning to human brains, that in our newer data, where we're starting to profile, we're going back to profiling all the different states of cells in the mouse brain and not focusing only on astrocytes, we see indeed that there is a disease-associated state across all the different glia cells. So here you can see a disease-associated state in astrocyte cells, microglia cells that I've mentioned before, and also oligodendrocytes. So this really points to the hypothesis I raised at the beginning, that the entire cellular environment of Alzheimer's is actually changing. And this is, we see in mouse model occurring very early during disease progression. So for today, I would try to first show you that all these findings in mouse models are in fact also recapitulated in human brains, even though we're transitioning to a different genetics and a slightly different form of the disease. And so when we transition from mouse models to human brains, our challenge is actually quite big, because we have a diversity of genetics, backgrounds, lifestyles, and different environments, which all of these are actually risk factors for Alzheimer's itself. But we used a similar strategy as we used in the mouse models by building cellular profiles of the healthy brain and the Alzheimer's brain using single nuclear RNA sequencing and comparing between them. So this is, again, a collaboration with multiple other labs. I'll just mention mainly Phil de Jager and Vilas Menon from Columbia University have been working really closely with us on this. And all the results I'll show you today are from Anael Kain, my student. So as I said, we profiled 24 individuals So these are post-mortem brains of the prefrontal cortex area of both healthy individuals and Alzheimer's individuals, both men and women, all are from aging populations. And this is the map that we've been building. You can see we identify all the different cellular populations that we expect to find in the brain, multiple different types of excitatory and inhibitory neurons, all the different types of glia cells, and so on. So we can now start to actually discuss the cellular diversity that we find in the human brain, in all the different characteristics of it. And similar to what we did before, we then focused on each of these different cell types and profiled the diversity of subtypes and cell states in each of these. I'll quickly kind of summarize a lot of data about the cellular diversity to reach the more interesting part about the disease relevance. When we look across all the different cell types, we can find a diversity of states in astrocyte cells, microglia, endothelial, and multiple subtypes of neuronal cells, kind of establishing the diversity of cells in the brain. Specifically, when looking at astrocytes and microglia, we see a transition from, again, a continuous transition from a more homeostatic state to multiple different disease states, and we can link them to the same cellular markers we found also in the mouse brain. So in some sense, we're finding some similarities to the mouse brain, only we have multiple reactive states, unlike the mouse brains. And when we ask which is the most relevant state, a reactive state to the disease-associated astrocytes I just showed you, we find similarities to the fibrous-like reactive astrocytes that are also upregulating immune and metabolic and stress response signaling. So we found that very exciting. I just want to comment that not all cell types could actually be easily characterized by using discrete methods like clustering. So we also used alternative methods like topic modeling that were recently shown in order to find expression programs that are unique to that are changing across all the different cells. And every cell we now have a soft clustering approach where every cell is modeled by a combination of expression programs. And here you can see the strength of the association of each cell to each expression program. So now that we have completed our math, we can really ask what are the disease-associated states in the human brain, and we can show some circumstantial evidence from 24 individuals showing disease association to astrocyte states or to specific programs in oligodendrocytes. But our major problem is our scalability, that within 24 individuals, we have very limited ability to connect between disease states and cellular states because of the huge diversity in the human population. So we actually used a different approach using bulk sequencing that was available for us from 640 individuals, and then using a deconvolution approach to predict in each of the 640 individuals what is the frequency of each of the cellular populations we just described using the single cell RNA sequencing. So now we have a prediction of the cellular landscape in 640 individuals, and we can now connect them to disease. So we could see, for example, cellular populations associated to a cognitive decline and to neurofilament tangles. So, for example, the oligodendrocyte programs I've just showed you, one of them strongly associates with cognitive decline, while another associates with cognitive resilience. And similar to that, you find microglia cells and others. So I'll just quickly, in the 2 minutes I have left, I'll just show you how we're now moving from single cells to entire cellular environments. So the idea is very simple, that when we look at all the cellular states that we can find in a specific cell type, when we look across 24 individuals or even 640 individuals, we see that they're not uniformly distributed, but rather specific individuals have mainly one state and not others. And you can see it here also in the 640 individual, where it's much more on a continuum of states. So we can now actually look across all the different cell types and see that in all of them, we find these interesting switches across cell states. And we can ask, are these changes actually coordinated across individuals? So we can look in combinations of different cell types and look if they're coordinated, correlated, anti-correlated, or maybe have no relation at all when we're looking across 640 individuals. So we're building these detailed pairwise correlation matrices to find these correlations across all cellular populations. And we actually really prefer to represent that using a graph where every specific node is a different population colored by the cell type, and they're either strongly correlated using the red line or anti-correlated using the dashed blue lines. So you could clearly see there is a pattern within our graph and matrices, and we can use a community detection algorithm to assign cellular populations to cellular communities. And each cellular community is more frequent in one individual and less in the others. So now we're actually divided the brain into cellular communities where different brains have more abundancy of different cellular communities. For example, we have a community of homeostatic astrocytes, microglia and endothelial cells, and others that have reactive astrocytes and reactive microglia cells. We could also try and now look at the signaling between them and find signaling that are specifically linking different cellular communities. But just to complete our story, I'll just show you how we're now linking astrocyte pathologies to our cellular communities. So we could look at the individual score for every cellular population to how much it's related to different Alzheimer pathologies or to generally cognitive decline, showing that we're finding a specific cellular population that's highly linked to all the Alzheimer pathologies and to cognitive decline. So we can now annotate our communities, showing we have a community related to amyloid beta, tau pathologies, cognitive resilience, and the homeostatic and reactive communities. So back to the cartoon representation, we're thinking that we have now the beginning of a description of how does the cellular environment of cognitive decline looks. So I'll just summarize. We started by looking at mouse brains, and defining glia-associated cells, transitioning to human brains, showing the same populations, and then to cellular communities that are linked to cognitive decline and resilience. And I'll just end by really thanking all my different collaborators and funding, and of course my lab, which is great fun to work with in COVID remote times and also when we're all together. So thank you for listening.
Thank you, Naomi. So we have a series of questions from people in the audience. So one of them is from Shauna Cooper, who asks, the penetrance of AD is nearly 100% in people with trisomy 21. Are you seeing changes within reactive cell types indicative of transcriptional changes that are chromosome specific?
Wow, that's a great question. We actually did not check the chromosomal bias, but we'll definitely do that and get back to you. It's a good suggestion.
Yeah, it seems like a cool idea. So Mario Flores asks, in what cases is it better to use topic modeling instead of clustering?
So that's a good question. So we haven't invented topic modeling, but I think we're really doing a good use of it. So when you have a cellular populations that can be described by discrete clustering approaches, where you have like fuzzy borders between clusters, this would be a good place to use any approach that uses more soft clustering. And topic models is one of these approaches where you have gene programs, and every cell is modeled by a combination of programs instead of just being assigned to one cluster.
Cool. Reagan Hamill has a question, and I actually have sort of a little bit of an addendum to this. So thanks for a great talk. Have you investigated the spatial aspect of these disease-associated astrocytes, e.g. proximity to plaques, microglia, and vasculature? And actually a question that I had, I think, related to this is, so you see a change in the expression profile of particular cells, and you see some, localization difference. Now they're closer to plaques, if I got it right. I guess one of my questions is, how much of the expression profile is intrinsically different? Like the cells are intrinsically now acting differently versus, well, now I'm near something else, and in response to being near the plaque, I have a change in gene expression. So in principle, it could be all one way or all the other, and I'm wondering if you have any
Right.
sense to which. But the primary question was about spatial aspect of these DAAs.
Yeah. So you actually said nicely that we did show that the disease-associated astrocytes are positioned close to a beta plaques. That's in the mouse brain. We're still exploring these aspects in the human brain using alternative methods. So we don't have, for example, the cellular communities that we found. We still need to show that they are or are not close to disease pathologies. But the question you raised about what is like an intrinsic change in cell state and what is driven by the proximity to pathologies, this is, I mean, of course, I don't have an answer for that because what we're measuring is how it is in the tissue, and it's hard to separate between these two. But it's definitely very interesting. But I can tell you what I think. So what I think is that it really depends a lot on the cellular and microenvironment you're at, which means the secreted molecules around you. And so are these molecules coming from blood vessels and would actually affect all the tissue simultaneously? Are these really locally driven by cells that are in proximity to plaques and you're in proximity to these cells? That's something we're still investigating. And I think this is one of the more critical questions, I would say, to ask what is the major signaling that are driving these changes?
Yeah, thank you. So apologies if I get this name poorly pronounced, but Mikhailo Batiuk asks, what is a proposed function of disease-associated astrocytes in Alzheimer's pathology?
Yeah, so we think that they actually have slightly different roles at different stages of the disease. At the initial stage, we actually think they might even be beneficial and definitely driven by the amyloid beta pathology. So that's our hypothesis. But at later stages, we think they actually are already neurotoxic on their own, and there is some evidence based on the pathways that they are expressing. And also, actually, some of these aspects are confusing because we've shown that there is a switch from the homeostatic state to the disease state. So just the lack of having enough homeostatic astrocytes to support the multiple functions that they have in supporting homeostasis and metabolism could be actually harmful for the brain on its own. So even though astrocytes might be important in responding to the pathology in the brain, just not having enough homeostatic astrocytes could be very detrimental. So we think that actually, while microglia cells are actually probably have a beneficial role at the beginning of the disease, the astrocytes will probably be the source of inflammatory and neurotoxicity in later stages of the disease.
Cool, thank you. So we will take a break now, and any unanswered questions will be available in the session forums. So, please feel free to interact with Naomi there. We'll be returning at 11:40 Eastern time, Eastern Daylight Time, I guess. Translate that to your time zone, and we'll see you in about 7 minutes. Okay, thank you all.
Thank you.